This conference will now be recorded.
Good evening, students. I'm Dr. Divya from TeenEinstein. So this is part 4, our fourth class of chapter 11, Biotechnology Principles and Processes. Okay, so in this chapter, we are learning about different biotechnological processes and principles behind those biotechnological processes. So we are learning about how we are making use of these technologies, these different biotechnological processes and principles behind those, how we are making use of these technologies in today's life, or how to make use of all these technologies. That's what we are going to learn in this chapter, and few techniques we have already learned in part 3. That means in last class we had discussed about the features of cloning vector which is required for cloning procedure. So a cloning vector should have some minimum features. What and all they, we have already discussed all these things in the last class we had discussed. So a cloning vector that need origin of replication. So what and all cloning vector need? Origin of replication. Then a cloning vector need a selectable marker. Then a cloning site that need cloning sites also. Then vectors for cloning genes in plants and animals. So the features that are required to facilitate cloning into a vector. A vector needs some kind of features, the minimum features which a vector required. So origin of replication, selectable marker, cloning sites, vectors for cloning genes in plants and animals. So these are the minimum features that are required to facilitate cloning. Okay, here you can see plasmid vector, bacteriophages, cosmids, all these are different kind of vectors which we are used for cloning. So let us discuss all these features in detail. We can discuss one by one. So the which are the features? Origin of replication, selectable marker, cloning site, vector for cloning genes in plants and animals. We'll go through all this. The first one, what we are going to discuss is origin of replication. Okay, origin of replication. The first feature, that is the first feature, origin of replication. Origin of replication, what is that? Origin of replication. Origin of replication is a sequence. It's a sequence. So it is a special sequence in that genome. Okay, here you can see, here we have marked as origin of replication. See, in this part, here we have marked as origin of replication. That is a special sequence in the genome. And in this this special sequence, or at this point, the replication procedure, it is getting started or it is getting initiated at this point. Okay, so that is called as origin of replication. From the term itself, it's clear. Origin of replication. That is a origin that at that point the replication is starting. So that is also a sequence in the genome. That origin of replication, that is also a sequence, and at that point the replication process is getting started. Okay, that is the origin. So within a host cell, any DNA fragment which is linking with that origin of replication, then along with that host genome, this DNA or this foreign gene, that is also getting replicated, right? Here, or the origin of replication, at this point the replication process is getting started. That is the origin. So when we are linking a DNA or a gene of interest, or any piece of DNA which is linked to that sequence, that can be made to replicate with that host cells, along with the host cell or along with that replication, this DNA or this gene of interest is also getting replicated. So that point where that replication procedure or replication process is getting started, that point is called as origin of replication. So origin of replication is the starting point of replication. We can say like that. And the sequence, we said that origin of replication, that is also a base sequence. Okay. So this sequence where it is acting as the origin of replication, that is responsible for controlling the copy number of the DNA or the foreign DNA. That foreign DNA copy number, it is controlled by the sequence of this origin of replication. Okay. So the sequence which is acting as the origin of replication, that is responsible for controlling the copy number of the foreign DNA, or to which that host DNA, the foreign DNA is linked to that host DNA, right? So to that vector DNA, it is linked. So that origin of replication, that base or that base sequence, that is controlling the copy number of that foreign DNA. Okay. So what scientists will do, if they want high copy number, then they have to select a vector which is supporting high copy number. Okay. So this sequence that is controlling the copy number, right? This origin of replication, that sequence is controlling the copy number. So if we want high copy number, we have to select a vector which is supporting the copy number, more copy numbers. Or if we want a low copy number, according to that also we need to select the vector. So while selecting the vector, there are some criteria to select the vector. So if we want more copy number, if you want high copy number, we have to select such a vector which is giving high copy number. Okay. If we want a very low copy number, then according to that we need to select our vector. Okay. So origin of replication means the sequence from where replication is starting. Okay. With that piece of DNA, that foreign DNA which is linked to the sequence, that can be made to replicate along with the host cell. So this sequence, that sequence is responsible for controlling the copy number. Which sequence? That sequence which is present at the origin of replication. Okay, that sequence is responsible for controlling the copy number of that linked DNA. So if you want more copy number, or if you want many copies of that target DNA, we should select a vector which is giving high copy number. If you want a low copy number, or we want very less number of copies of that linked DNA, we need to select the vector which is giving a very low copy number. Clear? Then next feature, we said 3 to 4 features of vector, cloning vectors. So the second feature is selectable marker. Now we are going to discuss about selectable marker. So first, what we said, a vector should have origin of replication, right? So the second feature is selectable marker. So this is for the selection of recombinants. We know that what is a selectable marker? A marker which is helping for the selection of recombinants. That is the selectable marker. So the second feature is selectable marker. So a selectable marker, that is to identify the transformed cells or the recombinants. Okay, and that's how we can make selections, and we can eliminate the non-recombinants also. So selectable marker, for what purpose we are using? For making selections. So with the help of the selectable markers, we can identify the transformed ones, and we can eliminate the non-recombinants. Okay, the selectable markers are helping to select the recombinants. These selectable markers, they are helping or they are supporting the growth of recombinants or the transformants, the recombinant one or the transformed ones. The recombination in which the recombinant DNA is present, they are called as transformants. Okay, so in transformants, how we can make out, with the help of the selectable markers, we can make out the recombinant and non-recombinant ones, right? So we can eliminate the non-recombinants or non-transformants which are not having gene of interest, that we can eliminate, because no need to select that. That will be the common cell only or the common vector one only or the common host cell which is having the gene of interest, only those we need to select. So for that purpose we are using selectable markers. So a selectable marker is helping to identify the transformed cells or the recombinants, and that is helping us to eliminate the non-recombinants. So this selectable marker, that is supporting the growth of recombinants. Okay. So in transformation, what is happening in transformation? A piece of DNA or that foreign DNA which is having the gene, right? The foreign DNA that is having the gene of interest, that will be introduced into a host bacterium. So usually antibiotic resistant genes, they are used as selectable markers. In normal cases, what we will use, antibiotic resistant genes, they are used as selectable markers. For which one? For E. coli. That means the genes which are encoding resistance to antibiotics, those genes will give resistance against antibiotics. For example, genes encoding resistance to chloramphenicol, then tetracycline, kanamycin, all these are mainly used for selection of antibiotic resistant ones. That means the recombinants. Okay, to select the recombinants. So mainly these are used as selectable markers in E. coli. Note down, in E. coli, what and all we will use as selectable marker? Kanamycin, uh, the genes which are encoding resistance to antibiotics. Which and all the antibiotics? Ampicillin, chloramphenicol, tetracycline or kanamycin. Okay. Then why we are using this antibiotic resistant genes as a selectable marker in E. coli? Do you know why we are using this antibiotics? Why we are using these antibiotic resistant genes as selectable markers in E. coli? What is the purpose of using, or why we are using these antibiotic resistant genes as selectable marker in E. coli? Because normally in normal cases E. coli cells, they are not resistant against antibiotics like ampicillin, chloramphenicol, tetracycline or kanamycin. Okay, normally in normal condition it is not resistant. E. coli cells are not resistant to these antibiotics, which and all? Ampicillin, chloramphenicol, tetracycline, kanamycin. Okay, when E. coli cells are exposed to these antibiotics, that may die. Okay, they can't resist. They don't have the resistant gene. E. coli cells don't have the resistant gene of these antibiotics. So if you're introducing an antibiotic resistant gene into an E. coli cell, that gene starts coding for the protein which makes resistance against the antibiotic. Right? If you're introducing a antibiotic resistant gene, like antibiotic resistant gene, which and all these mentioned antibiotics, not for all, Okay, when we are introducing any of this, an ampicillin resistant gene or chloramphenicol resistant gene into an E. coli cell, that gene, then it is a part of that E. coli, then that gene will code for the protein, and that makes the resistance against the antibiotic, right? So that is having antibiotic resistant gene as a selectable marker, right? When the cells, all these cells which are having antibiotic resistant genes as a selectable marker, when they are growing in a medium which is having antibiotic. See, suppose we are introducing an ampicillin resistant gene into E. coli, then that gene will work in E. coli. It will code for a protein and that will give resistance to ampicillin, right? When those E. coli cells, when we are introducing those E. coli cells into a medium which is having ampicillin in that, anything will happen to that E. coli cells? No, because it is having ampicillin resistant gene in it. Okay, those E. coli cells that we have already introduced an ampicillin resistant gene into that. So when we are making them to grow in an ampicillin, a medium which is containing this ampicillin, in E. coli cells, these ampicillin resistant gene which we have introduced, that will start working. It will produce resistance against that ampicillin or that antibiotic. So only those one which are having that resistant gene can grow in that medium. Other, the remaining ones which are not having the resistant genes, that may die or that cannot survive in that medium because that medium is having antibiotics. Usually, how these antibiotics will work? Antibiotics, what it will do in normal cases, antibiotics will work by blocking that vital process. Okay, in bacteria, they'll block the vital process or they'll kill the bacteria or they'll stop them from the multiplication. They will suspend or they'll stop the multiplication of bacteria. In any of these ways, they'll block the growth of the bacteria. So when E. coli, which is a bacteria, it is having antibiotic resistant gene that is having the capacity to resist that antibiotic or resist against that antibiotic, right? So when we are introducing such kind of bacteria or E. coli, which is having that antibiotic, specific antibiotic resistant gene in it, when we are introducing such E. coli into a medium which is having a particular antibiotic, definitely it will show the resistance. So those who can survive, they are the recombinants. Okay, they have that introduced gene in it. They are the recombinants. Others will die, right? So when it is having the resistant gene in it, it can grow. That means only recombinants or the transformants can grow. Others may die or they cannot survive. That is the common way of working, right? So the normal E. coli cells, they won't carry resistance against that antibiotics. Okay, so that's how it is working as a selectable marker. So a selectable marker, how these selectable markers are working or what is the role of that selectable marker? They are helping for the selection or they are marking the recombinant ones. Okay, so the genes which we are using normally as selectable markers? Antibiotics. Which and all the antibiotics? Ampicillin, chloramphenicol, tetracycline and kanamycin. These are the common antibiotics which are used as a marker for E. coli, selectable marker for E. coli. Because normal E. coli cells, they do not carry resistance against any of these antibiotics. Which and all antibiotics? Ampicillin, tetracycline, kanamycin and chloramphenicol. E. coli cells in normal cases, they don't have resistance against these antibiotics. When we are introducing a gene of interest into these E. coli cells, that means a gene of resistance. This antibiotic resistant genes, when we are introducing to E. coli, that will show the resistance. So when we are adding these E. coli cells into a medium which is containing that antibiotic, any of this antibiotic, that then it will show the resistance, and the resistant ones or the recombinant ones can survive. Non-recombinant ones will die because those antibiotics will block the vital process, or it will block the growth of that bacteria which is not having the resistant gene in it. Okay. So here, how we can use as a selectable marker? The one which is having antibiotic resistant gene can survive. So we can make out they are the transformants, right? And that helps in the selection also, because normal E. coli doesn't have antibiotic resistant gene, right? Then, for example, PBR322, which is a E. coli cloning vector, PBR322. Here you can see PBR322, which is a vector, and it is having two selectable markers. These selectable markers, they are antibiotic resistant genes for tetracycline and ampicillin. Here you can see this is PBR322. We have to learn the structure of PBR322 or the features of PBR322, and it is a question for your board exam mainly, and from this you may get MCQs like the selectable markers which are present in PBR322. Okay. And here you can see tetracycline here. Tetracycline, this is the selectable markers. Here you can see tetracycline resistant, TTR, tetracycline resistant and ampicillin resistant. These are the selectable markers which are present in PBR322. PBR322, which is a vector, it is having two selectable markers, and these are the antibiotic resistant genes, which are the ampicillin resistant one, then tetracycline resistant. The resistant genes for ampicillin and tetracycline. So these are the selectable markers which are present in PBR322, which is a E. coli cloning vector. Okay, PBR322 is a E. coli cloning vector, and it is having two selectable markers which are antibiotic resistant genes. Which are those? Ampicillin resistant and tetracycline resistant. These are the two genes, antibiotic resistant genes present in PBR322. Don't get confused. Again, from this you have learned one feature of PBR322. It is having two selectable markers which are antibiotic resistant genes, ampicillin resistant gene and tetracycline resistant gene. Okay, so here by learning the same, like about cloning vectors, the features of cloning vectors, you are learning about PBR322 also. One by one we can see. Okay, so the antibiotic resistant genes, ampicillin resistant gene and tetracycline resistant gene. Here you have to draw the picture of PBR322, and now one part you have learned, right? In that two selectable markers, ampicillin resistant gene and tetracycline resistant gene. Two antibiotic resistant genes are there. Two selectable markers are there. They are the selectable markers, ampicillin resistant and tetracycline resistant in PBR 322. Repeatedly I am saying because these questions are sure for your board exam. Next, now we have learned about origin of replication, then selectable markers. The third feature, third is cloning sites. Okay, next feature is cloning site. So a cloning vector should have cloning site. So what is that cloning site? Cloning site means the site where DNA is inserted into a vector. So here you can see origin of replication, then ampicillin resistance, tetracycline resistance, two resistant genes you can see. Next, what we said, cloning site. Okay. So what is that cloning site? Cloning site means the site where DNA is inserted into a vector. The site where that DNA is inserted into a vector. At that site, the foreign DNA or the alien DNA is getting inserted into that vector. So we have already discussed about restriction enzymes, right? We have learned how restriction enzyme will work, how it will cut. So the restriction enzyme that recognizes the recognition site. Okay, that recognition, see this restriction enzyme that can recognize the site where it want to make the cut, right? For the insertion of the foreign DNA, it will find out where that need to get attached. Then we know that it will cut not exactly at the site, little away, and it will make sticky ends, right? That and all we have learned. So for linking that alien DNA or foreign DNA, a vector should have very few recognition sites. Okay, so a vector should have very few recognition sites. So for commonly used restriction enzyme, they should have very few recognition sites. So mainly we will say that, or preferably we can, single recognition site is better. We can say that single recognition site is better. Why we are saying like that single recognition site is better? Because if more number of recognition sites are there means that will create confusions and it will make lot of fragments, right? See, if one recognition site is there, they only there the restriction enzyme will go and bind, it will make cut, right? If there are many number of recognition sites, everywhere it will go and bind and it will make so many fragments, right? So that may create confusion and complications in the gene cloning. So here in this picture you can see PBR 322. Here again we are saying about PBR 322 and PBR 322 echo R1, then BAM H1. These are the recognition sites. So we have learned in the previous one, we have learned about it is having two selectable markers which are antibiotic resistant genes. Okay. Next we are learning PBR 322. Another character, another point about PBR 322. Here PBR 322, it is having restriction sites. Here you can see restriction sites. Which are they? Echo R1 and BAM H1. So these are the restriction sites. Here you can see antibiotic selectable markers. Then Echo R1. This is the recognition site. Echo R1 and BAM H1. Okay. So these are the recognition sites. Echo R1 and BAM H1. These are the restriction sites. There the restriction enzyme will come and attach. Okay. So restriction enzyme that will recognize that site and it will come and attach there, and it will make cut, and thereby making the sticky ends, right? For what? For attaching, for the insertion of the foreign DNA. So for linking that alien DNA or that foreign DNA, a vector should have some recognition sites. So without cutting, we can't insert that DNA anywhere, right? We need to make a cut in that vector. Then only we can insert the foreign DNA or our gene of interest. So for that, making cuts, a vector should have a recognition, a site, a restriction site. Okay, so that cloning site should be there. So in that cloning site only, this restriction enzyme can attach and make cut, right? That recognition site is must. Recognition site only that restriction enzyme can attach. So that a vector should have few recognition sites. So what we can say, single recognition site is best. Why? Because if they are having more than one recognition site, everywhere this restriction enzyme will go and bind, and everywhere it will make cut, so that more fragments, that will make several fragments. So that may create complication in gene cloning. Okay, so in PBR 322 here, for example, we are saying about cloning site. We are, for an example, we are saying about PBR 322. In that, BAM-H1 and ECOR1, these are the recognition sites, or these are the cloning sites. These sites, that restriction enzyme will come and attach, and it will make cut. Okay. Next, this ligation of that foreign DNA, that will carry out at a restriction site present in one of the two antibiotic resistant gene. Okay, here that restriction enzyme will come and bind, and it will make cut at any of these sites. Okay, so at this site where that antibiotic resistant genes are present, there only it will come and join. The ligation or the joining process of that DNA, it is taking place at a restriction site which is present at any of the antibiotic resistant genes, or the place where the antibiotic resistant genes are present, at any of these place, right? Here, example, ligate a foreign DNA at the BAM-H1 site of tetracycline resistant gene in the vector PBR 322. Okay, here, see here, this recognition site, it is getting that restriction enzyme is getting attached, and it is making cut here, so that it is making sticky ends here. Then the foreign DNA or the alien DNA is getting attached in this site. So it is having tetracycline resistant gene in it. Okay, so that will be having, that recombinant will be having tetracycline resistant gene in the vector PBR 322. Okay, so the otherwise if it is getting attached here, it will be having ampicillin resistant gene, right? So when it is getting attached, see here they have marked as the recognition site, and here tetracycline resistant gene is present. So when it is getting attached here at this site, it is getting recognized and it is getting attached. The restriction enzyme is getting attached at this site which is having that BAM-H1. Then that will be having tetracycline resistant gene. In that vector, it will be having tetracycline resistant gene in PBR 322. Got it? Then here we said in PBR 322 vector, we can ligate the foreign DNA at BAM H1 site for tetracycline resistance. Right? For getting tetracycline resistant, we can attach the foreign DNA at this site where the BAM H1 is present. At this site, we can make cut and we can insert the foreign DNA here so that it will be having tetracycline resistance. Isn't it? Then due to that insertion, when we are inserting that foreign DNA, due to that insertion, that recombinant plasmid that will lose the tetracycline resistance. Okay, here you know that tetracycline resistant gene is present here, right? Due to that insertion, then after making cut here, that foreign DNA is getting inserted into this point, right? Where this BAM H1 recognition site is present. At that point, we are inserting. Think like that. Imagine like that we are inserting here. So due to that insertion of that foreign DNA, that may lose, or this PBR 322 vector that may lose that, or that recombinant plasmid will lose the tetracycline resistance, that resistant power that may lose. Okay. Then how to make selection? Because that is a selectable marker that is working. This tetracycline resistant gene that is working as a selectable marker. So when we are inserting, due to that insertion of that foreign DNA, that may lose the resistance, tetracycline resistance. Then also we can make selections. How we can make a selection? We can plate them in a tetracycline containing medium. Okay, then we can select the recombinants from the non-recombinants. In a tetracycline containing medium, we can plate them. Right? Here, sometimes the recombinant plasmid, they will lose the tetracycline resistance due to that insertion of that foreign DNA. Then also we can separate the non-recombinants and recombinants. How we can separate? By plating that transformants on tetracycline containing medium. Okay, a medium which is containing tetracycline medium, a tetracycline antibiotic, on that we can plate them. Okay, by that plating method, we can make out the transformants. Then the transformants growing on ampicillin containing medium, they are then transferred on a medium containing tetracycline. Okay, then we are transferring them to the medium containing tetracycline. Then that recombinants, they will grow in ampicillin containing medium, but on tetracycline containing medium, they won't grow. Okay, but non-recombinants, they can grow on medium which is having both the antibiotics. Okay, non-recombinants will grow on the medium which is having both the antibiotics. Okay, but the recombinants, they will grow selectively. Got it? So we can say that, or we can conclude like antibiotic resistant genes, they are working as selectable markers and they are helping to select the transformants. But other antibiotic resistant genes, they will get inactivated due to this insertion of foreign DNA. When this foreign DNA is getting inserted, due to that insertion, sometimes they will lose the power of resistance. Okay, and that helps in the, then also we can select the transformants or the recombinants. Through the plating method, we can select the recombinants. So mainly what we can say that this is a proof how these recombinants, how selectable markers are working to make out the recombinants, or how, what is the role of selectable markers, or how they are working, or how they are helping to select the recombinants and non-recombinants. Okay. The next, see, from this topic you will get, mainly you will get application type question for your NEET exam, because they may give so many antibiotics, name of so many antibiotics, and you are inserting one vector and it is having a resistant gene, like that application type questions you may get. So you have to understand very clearly, you have to understand the concept of this features, or how these features are helping, or what is the role of all these features, how they are, how a selectable marker is working, how a cloning site is working, uh, restriction site, how it is working. All these things you should have a clear idea. Then you can think in a way that there, without confusion, you can answer everything. Okay. So concentrate on these topics. Mainly you have to get the concept. It should be very clear. Otherwise you cannot answer. You will get confused after reading the question. So next, selection of recombinants. See, just now I said it will get inactivated due to the ligation, or insertion of the foreign DNA and ligation of that antibiotic resistance power, that will lose in some cases. So the selection of recombinants due to that inactivation of antibiotics, it's a heavy task, or it is a difficult task, very difficult task, because in that cases we need plating on two places which is having different antibiotics. Here now we said ampicillin and tetracycline, how to make selection. So simultaneously we need to do the plating. Both plating we need to do with different antibiotics, right? So it is a very difficult task. It's not so easy to make out. So an alternative selectable marker that have developed, which is able to differentiate this recombinants, that will be able to differentiate the recombinants from the non-recombinants on the basis of their capacity to produce the color. Okay, in the presence of a chromogenic substrate which is creating, which is making color when it is getting reacted. So at, how we can select the non-recombinants on the basis of their capacity or their ability to produce color in presence of a chromogenic substrate. See, the plating method, simultaneous plating to find out the recombinants and non-recombinants, it is a difficult task. So we should have an alternative method. So for that, scientists found out some alternative method, which is a selectable marker, which is developed, or which can differentiate the recombinant and non-recombinant on the basis of their ability to produce colors when they are getting reacted with a chromogenic substrate. Got it? See, for an example, see how that chromogenic substrate is working. For that, we are saying an example. Here, a recombinant DNA is inserted within the coding sequence of an enzyme beta galactosidase. Okay. So within the coding sequence of an enzyme beta galactosidase, a recombinant DNA is getting inserted. Then when we are inserting, due to that insertion and due to that ligation, that gene that will result in the inactivation of the gene for synthesis of this enzyme. And that kind of inactivation, due to the insertion, some genes are getting inactivated and they are not able to synthesize the enzyme. Such cases, it is called as insertional inactivation. That is called as insertional inactivation. Then the presence of a chromogenic substrate that gives blue colored colonies if the plasmid in the bacteria does not have an insert. Got it? The presence of that chromogenic substrate. If that chromogenic substrate is present, then it will give blue colored colonies. When it is getting reacted with that chromogenic substrate, it will give blue colored colonies. When, if the plasmid in that bacteria does not have an insert, if there is nothing is inserted, or there is no recombination, or there is no, that is not a recombinant one, or there is not foreign DNA in that, in such cases it will give blue color. Okay, when, because if that is not getting inserted, it is not losing the power. That enzyme is getting synthesized, right? The enzyme synthesis is happening, everything will be common as usual. But when an insert or something is getting, or foreign DNA is getting inserted, it is losing the power to synthesize that enzyme. Then when it is reacting with the chromogenic substrate, it won't be able to produce any colors. Without an insert, it is having the capacity to produce the enzyme, or it is not losing, or it is not getting inactivated. Okay, insertional inactivation is not happening. If it is not happening, it will produce that enzyme, and the presence of chromogenic substrate, when that enzyme and substrate reacting, it is getting reacted, it will give blue color, blue colored product. But when something is inserted into that plasmid, or the gene of interest is inserted into that plasmid, it will lose the power to synthesize that enzyme. Means insertional inactivation is happening there. It is getting inactivated and that enzyme is not getting produced. If that enzyme is not there, it won't work with the substrate. There won't be the production of any enzyme substrate complex in blue color. So when the insert result into insertional inactivation of beta galactosidase gene, and that case there won't be any enzyme substrate complex, they do not produce any blue color. Okay, so these are the blue colored one, they are the non-recombinants, and the blue without any color, they are called as the recombinant colonies. So that's why with the help of color we can make out the recombinants and non-recombinants. So this is an alternative method which is used. Okay, when they are losing the power, the antibiotic resistant genes, when they are losing the power to select, or there is an insertional inactivation is happening, in that case we can find out the recombinants and non-recombinants in this way. Okay, when it is getting reacted with a chromogenic substrate, it will give blue color. So when there is an insertional inactivation, that enzyme won't get produced, that inactivation of beta galactosidase gene, and thus the colonies will never produce blue color or any color. It won't produce, it will become colorless, and that will be identified as recombinant colonies. So that's how we can identify recombinant colonies. Next, last we said, now origin of replication over, restriction sites over, then which one we said, selectable markers, that one over. The last one, vectors for cloning genes in plants and animals. Okay, fourth point we said, vectors for cloning genes in plants and animals. So how to transform eukaryotic cell, or how we can deliver a gene to eukaryotic cell. Up to this time, we were talking about the prokaryotic. In bacterial cell, how we are delivering, or in to E. coli, which and all vectors we are using. In that way, we were talking. It was easy, right? But in eukaryotic cell, in the case of eukaryotic cell, how we can deliver a gene into a eukaryotic cell? See, there are several methods for delivering genes into eukaryotic cell, like agrobacterium tumefaciens. Means how to deliver in plant cell and animal cell. Different types of methods are there, different kind of methods are there. For example, agrobacterium tumefaciens. That is, you know that agrobacterium tumefaciens, it is a pathogen of many dicot plants. Okay, it is infecting so many dicot plants. Actually, it is a pathogen. So this can deliver a piece of DNA. This agrobacterium tumefaciens, this just for an example, this is a vector. Just for an example we kept here. Same like this, how vectors, we can give vectors for cloning genes in plants and animals. That's what we are going to discuss here. So agrobacterium tumefaciens, that is actually a pathogen, and that is infecting so many dicot plants. Okay, so by making use of this agrobacterium tumefaciens, how we can transfer or how we can deliver a piece of DNA into plants. So this agrobacterium tumefaciens that can deliver a piece of DNA or a foreign DNA into plants, that is called as tDNA. That tDNA to transform normal plants into a tumor. Okay, they can transform normal plant cells into a tumor and direct these tumor cells to produce the chemicals required by the pathogen. Okay, then these tumor cells, they can produce the chemicals which is needed for that particular pathogen. So in same way, in animals which one? Retroviruses, they have the ability to transform normal cells to cancerous cells. Got it? So in agrobacterium tumefaciens, so vector in plants and animals. In plants, agrobacterium tumefaciens, actually it is a pathogen. In retroviruses, these are also pathogens, but with the help of these pathogens, how we can transfer a gene of interest or a foreign DNA into plants and animals. That's what we need. How to make use of these pathogens to deliver the DNA, which or the gene of interest, into the eukaryotic cell. We said agrobacterium tumefaciens and retroviruses, both are pathogens. So scientists, they got an idea about delivering genes. See how we are inserting this, how we are making use of this vector. We are finding out a restriction site, then we are inserting the foreign DNA, and this vector, it is transferring that foreign DNA into a host cell. So in the same way, how we can make use of a plant pathogen on animal pathogen, and how we can transfer the DNA. Okay, so scientists, they got a very clear idea about the deli delivery of these genes by making use of these pathogens. So these pathogens will deliver this gene of interest into that eukaryotic host. That means in which one, or in plant or animal, whatever may be, in which one it is causing that disease, to the same eukaryotic cell, with the help of this pathogen, they are delivering the gene of interest. Got it? to the same, to which one it is causing diseases, to that one, to that cell, with the help of these pathogens, delivering the gene of interest. So here they transform these pathogens to useful vectors. So here these pathogens are working as useful vectors. Here how these plasmids, bacteriophages and all working as useful vectors. In the same way here, the animal pathogen or the plant pathogen, that is working as a vector or a carrier for delivering the gene of interest into humans. So in the case of TI plasmid, that is a tumor. TI means tumor inducing plasmid. So that is a tumor inducing plasmid of agrobacterium tumefaciens. So that has been modified into cloning vector. Okay. So that pathogenic effect, that is removed. That pathogenic effect is removed. Instead of that, that tumor inducing capacity, that is being used. Okay. But this is used to deliver gene of interest to many plants. So they are using the same, how that pathogen is infecting a plant, the same mechanism is being used, but the pathogenic effect is removed, that is modified. Okay. So these plant pathogen or TI plasmid, tumor inducing plasmid of agrobacterium tumefaciens, how it is getting infected to a plant, how a plant is getting infected with this pathogen, in the same mechanism. The same mechanism is being used for transferring the gene of interest. With the help of this tumor inducing plasmid of agrobacterium tumefaciens, the gene of interest is getting transferred to that plant. So the same infecting mechanism is followed, but the pathogenic effect is removed. Okay. So the retrovirus, in the case of retrovirus also, it is disarmed. Means that pathogenic effect will be removed and they are now used to deliver desirable genes into animal cells. So when one gene or a DNA fragment that is ligated to this suitable vector, that is a retrovirus or TI plasmid, whatever it may be, it is transferred into a bacterial, a plant or an animal host, and there it is getting multiplied. Okay. When it is transferred into a plant cell or animal cell or a bacterial cell, it is getting multiplied there. Got it? So up to that, hope it's clear. Now we have some more topics. We have to learn the features of PBR 322. I'll explain with structure. I need to explain the PBR 322 and some more points in this chapter. So hope up to this everything is clear. This is a confusing chapter. If you are not able to understand the concept clearly, you'll get confused. So make it clear. Hope up to this everything is clear. You have any doubt means you can ask me in the next class. Thank you.